So we're lucky to have a great connection out here in Malawi. So currently I'm in the southern part of Malawi at the moment, and we've got 5 students with us this year. Some are out in the community and some are here working in the laboratory, but we'll come to those later on. I'm the Director of Studies for the Tropical Disease Biology Programme for the MSc programme, and this programme largely takes the principles of biology in the widest sense, and we apply those principles of biology to the field of tropical disease. So a lot of what we look at are those endemic diseases that we often find across that tropical belt of the world, where the unique biology of that climate and that region often suit those particular parasites, microbes, and those vectors that spread those particular parasites and microbes. It suits their biology, but also across that tropical belt of the world, there are various regions that may be within low and middle income countries that also face particular challenges with their health systems, with their sanitation and hygiene aspects, and these exacerbate many of the diseases that the School of Tropical Medicine study as well. So those 2 main aspects provide us as biologists an opportunity to look at that unique biology and those unique challenges and to see how we can apply those themes. And those themes may be from a morphological, zoological aspect in humans and animals as well, so not just humans. Some of these diseases are zoonotic as well. It also allows us to apply some of those more modern molecular techniques and biochemical and cellular biology techniques. It also allows us to apply some of those in silico scientific computing methods as well. So we take aspects of field biology, laboratory biology, and in silico computing biology as well, and we put all those things together as a range of disciplines in order to tackle tropical diseases. So many of the techniques that you learn within tropical disease biology are also applicable to a range of fields of biology and investigation and research as well. So whilst it might sound quite niche, tropical diseases, all the applications and the techniques and the skills that we utilize during the year with us are also applicable to, say, cancer research, environmental research, animal and human research in a range of diseases and non-communicable diseases too. So it really does set your CV up for not just tropical disease, but a range of other diseases as well. The semester starts off in September of every year and runs through to the following September. We start off as soon as you come into the school. Within induction week, we take all the class off to Shropshire, which is within England. It's about 70 or so miles south of Liverpool, to a beautiful county called Shropshire. And in fact, the field centre we take everybody to for 3 days is just down the road, literally a mile down the road from where Charles Darwin was born. So it's a perfect place for biologists to come and convene and get to know one another. But also during the 3 days, we collect and study a variety of the temperate parasites and microbes and vectors of disease. You'll study the tropical cousins of those and during the rest of the year as well. So it's a great introduction, not only to the biology of what we're going to look at through the year and those collecting techniques, but also a fantastic opportunity to meet your peers and for us to meet you and you to meet us as well, and really get the year off to a great start. Once we're back from that field course, those few days, we'll be a very tight-knit community of a class by then. We undergo introduction modules for the first 6 weeks. So you look at all the different bugs and nasties that we study at the school and the different types of diseases. This helps you decide what might be your favorite parasite or microbe or vector of disease. And during this time, we'll talk to you a lot about particular projects that might be on offer at the school. Some 150 or so have been on offer this year across a range of different diseases and organisms. You'll get a feel for what you kind of like then, and you'll begin talking to different supervisors in order to pick a project ready for the following summer as well. After that first 6 weeks of introducing you into those, we talk to you about the different methods that we might use to study and collect and sample and investigate and analyze all those different organisms in a biological sense. So you'll learn a little bit of basic stats, some scientific computing, some communication skills, how to read papers, and some laboratory skills. So you might be very familiar with some modern molecular techniques and microscopy, etc., or you might not be. So we kind of get everybody to the same kind of level by the time we get to Christmas with methods and with the organisms we study. In the second semester, there's a range of modules that you can choose from, and by now you'll have got a feel for whether you want to follow a more laboratory molecular aspects, whether you want to follow more field biology aspects, or whether you like the more in silico scientific computing side of things. And your project will probably steer what modules you choose as well, in order to build up the skills necessary to do a wonderful dissertation in the summer. So those second semester modules, they run a run of 3 6-week modules, and they follow on with on top of one another, and they bring you to the end of May, and you'll have done those 3 modules there. By the time you get to the end of May, you'll have been discussing with your supervisor and chosen a project by now, and then from the end of May all the way till the first week in September is some 16 weeks or so, during which you'll do your project. And it may be that you'll come out to a field centre with one of our many hubs. I'm in Malawi, for example, at the moment. We've hubs in Kenya, Cameroon, Uganda, Zimbabwe, various places. But we've also got lots of collaborations around other parts of the world as well. Different departments have different grants running. So we've got some over 70 countries we've got a footprint in. It may be that you are more interested in laboratory projects, in which case you'll work within our state-of-the-art laboratories in Liverpool, or sometimes with our partners in the UK, some of them industrial, and some of those other institutes as well that we partner with. Many projects are collaborative these days as well. So we've got students working with Knowsley Safari Park, with Syngenta, with Manchester University as well. So that reflects the many partnerships that we have as well. But most of our students, if they do a lab project, would be in Liverpool typically. There are also field projects within the UK, such as in the safari parks, within zoos. For instance, last year I took a student collecting ticks all around the Lake District for the summer. That was a very nice project as well. So there's lots of UK-based projects, not just overseas as well. It may be that you want to do a scientific computing project. You may want to look at bioinformatics data. You may want to look at statistical data and health records. There may be meta-analyses you wish to do, etc. as well. So it might be that with a huge amount of big data coming for your future, you want to build up your CV and build those scientific computing methods that are going to be highly sought after in the future as well. So a whole range of different projects. And during the year, there are various conferences, for example, the British Society for Parasitology. This year, we've had several students join in as volunteers within that as well. There's lots of social activities take on through the year. There are lots of seminars you can go to, journal clubs as well. So there's lots of extracurricular and additional aspects that you can get involved in as well. And we also like to keep in contact with our students afterwards, and some of our students come and present their summer projects that they've done at events like the RSTMH Research in Progress, which is in December, just before graduation, for example, as well. So once you're with us, we like to help you show off your hard work that you've done during the year at various opportunities. It builds your CV and shows you off to various employers or other institutes for PhDs, for example. And we've been very lucky in retaining some of our previous MSc students, a couple of which I'll introduce you to soon here, that are sitting right opposite me in this makeshift laboratory that we've got here as well, that stayed on with the school as technicians and are now starting PhDs with us as well. Unless there's any questions, I'll begin to show you around the lab. So I'll just turn the camera away from me a little as I take you around the laboratory. we're in the southern part of Nasanji in Malawi, and we've been there's a local lodge that we've worked with for several years, and they've allowed us to use their conference suite and set it up as a laboratory. So I'll just show you that now. I hope you can see okay on this view. So I hope you could see that. I hope I didn't wobble it about too much. But as you can see, we've got lots of tables. We've set them out with microscopes, and all the microscopes are donated. Oh, thank you for the thumbs up. I'm glad you could see that. Good. And we've just got a delivery of samples. So some people are out in the community and have yet to come back. But within an hour or so, this will be really full of a whole team of us analysing samples. In this case, there are lots of urine samples. We're looking for schistosome eggs in those as well. And then we'll start hatching some of those eggs out. We've also got fecal samples, and we're looking for worm eggs in those as well. And we'll be archiving several aliquots of those material ready for molecular analyses back at LSTM, and also back in Blantyre, where we're helping build the capacity with molecular techniques there as well. If I could probably just take you over to a couple of our previous MSc students now who've stayed with us in technical roles first of all, and are now just entering their PhDs. So if I could introduce you to who's most ready to be introduced. Is it Ruth or Sam first? So I'll just show you. So this is Ruth and Sam. I think maybe they can hear. So Ruth and Sam are hatching Miracidia from eggs. Sam, I don't know if you want to say a few words. Feel free to grab that if you want.
Sound?
So yeah, about your journey.
Yeah, so I did the undergraduate degree, the BSc, and then I moved on to the MSc. It was a logical step, and I really like parasites because, and then from there I loved the work so much that I thought try and stay on. So now I work with Russ Zothard in the as a technician in his group, hopefully moving on to do a PhD part-time as well with the group. So it's yeah, I mean I can endorse it enough to say that I've stuck around for 8 years now, so it's not that bad. I'll give you back.
Thanks so much, Sam. Yeah, and I didn't have to pay Sam to say all those nice things. So Sam's been out to Malawi a lot in the last couple of years since he joined the group. I would pass you on to Ruth, but Ruth just started a procedure that I can't stop her while she's going on there, but I'm sure she might be able to give you a quick wave or something. So yeah, so I'll tell you about Ruth in her absence of being able to grab the phone and talk. So again, Ruth is processing samples that are coming from the hatching now as well. Ruth also did the MSc in tropical disease biology a couple of years ago, and then came to work in the insectaries, and then on into the education lab, and is now just starting a PhD in a month or two with LSTM as well. So Ruth's been on quite a journey within the school as well, but we're delighted to keep hold of Ruth as well. And Ruth, I think you did a laboratory-based project. Yeah, so Ruth did a laboratory-based project as part of her MSc, and Sam was meant to do an overseas project, but Sam's project was hamstrung by COVID, unfortunately. But he still managed to come to Malawi in the end as soon as COVID had finished, and he joined us as in a technical role at that point. I'll just show you another one of our current students. So this is Abby. So I'll pass the phone over to Abby if that's okay. Abby, are you free to chat very quickly about your journey and
Hi everyone.
What you're doing?
Yeah, so I'm Abby. I actually just finished my undergrad this summer. I was in the University of Liverpool. I did tropical disease biology, but I actually did my project at LSTM. So then I've switched over fully and I've started my master's now. So that's at LSTM, and then that's where I've joined the Hugs group. So yeah, so been doing research here for about 9 months now. So yeah, and now first time in Malawi.
Yeah.
Going good.
So what have you been up to this morning, Abby? Yeah.
So we went out to the local community today. I was just observing some like sample collection. So we were looking at collecting some urine samples. I also got to see some of the team working with the bloods, which was nice. And then some of the groups doing female genital and male genital schistosomiasis. So now I've come back, and this is kind of my area in the lab, working with all of our urine samples. So this is actually my helper. no, that's my boss. Putting on all of our tests now. And then we'll just follow all our samples along. But yeah, that's if there's anything else.
Thank you, Abby. That's great. Thanks very much, Abby. That was brilliant. And then lastly, but not least, we've Russ. So this is Professor Russ Stothard. Russ is leader of the team here in Malawi. Along with a Malawian colleague, Jane Lisa Masaya, who's not with us at the moment, but is meeting us in Mangochi on the next stage. So, Russ, I don't know if you want to say just a couple of words.
Oh, yeah. Well, welcome to the Liverpool School. As you say, I worked with James very closely for the past 12 years, and we try to integrate a lot of experiences in the field. That's the epidemiology, so looking at disease control into the teaching. So things like projects are very applied. We're really fortunate today because we had the National Control Program Manager making a site visit, and we were telling him, the findings as they're coming off from the results sheet. So he's particularly interested. So there's a really nice interplay between the research, teaching, public health policy, and some of the practices. And I'll just show you something. I've been uncracking up these tests. We're doing a batch testing of about 150 urines from patients, and then something which you don't see every day. It's a urine sample. I don't know if you can see the color of it, but it's not the right color, it's red. So that's a classic sign of urogenital schistosomiasis. So as Abby was saying, there's a sizable portion of the community here. About 50% have got infection, and about a quarter of them, about 25%, have got genital tract disease. That's in adult women and also adult men. So female and male genital schistosomiasis is really important. Any questions you can ask James, but we're a friendly bunch, and yeah, it's nice to work with the students. And you say, I'm the chief technician for Abby, working side by side, so pass them to James.
Thanks very much, Russ. Yeah, it's kind of true. Russ and I do become the technicians for students. We bring them out, settle them in, and then we basically become they boss us around for a few weeks while they collect their samples. So it's a fantastic experience, and we have a great time and plenty of fun while we're working hard as well.